Hello, everyone, and welcome to today's webinar, Approaches for Rapid Genomic Surveillance to Identify SARS-CoV-2 Variants. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by DiaSorin Molecular. To learn more, visit them at molecular.diasorin.com. Now, we want to encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions will be answered via email following the broadcast. You may also submit any technical issues here as well if you are having any trouble seeing or hearing this presentation. I'd now like to welcome our speaker, Dr. Donna Wolk, Division Director of Molecular and Microbial Diagnostics and Development from the Department of Laboratory Medicine at Geisinger. For a complete biography of our speaker, please visit the presenter information top of your screen. Dr. Wolk, welcome. You may now begin your presentation.
Hello. I'd like to welcome the participants to the webinar today. My name is Donna Wolk. I'm the Division Director for Molecular and Microbial Diagnostics at Geisinger Diagnostic Medicine Institute. I'll be talking to you today about the potential impact of SARS-CoV-2 variants on our laboratory testing. By way of a little background of where I work and live, the Diagnostic Medicine Institute is in Danville, Pennsylvania. We have 125 CLIA certified sites. We have 42 couriers and about 1,100 employees and about 15 million tests annually. And this is the region that we cover in central northeast Pennsylvania. So that's the perspective I'll have because I work here. The objectives today are to review the common molecular viral targets used for patient testing of the SARS-CoV-2 virus and list the emergency use authorization methods, discuss the potential of SARS-CoV-2 on the variants on the EUA test method, and describe different methods for variant detection. So first of all, let's start with what is a SARS-CoV-2 variant and why should I worry about it? Well, it does have implications for public health, the diagnostic industry and bioindustry, and the clinical diagnostic laboratory. The genes within the SARS-CoV-2 are listed here in the picture, the ORF genes, spike, envelope matrix, and nucleocapsid. The diagnostic tests we use in the clinical laboratory generally have more than one gene target, and that's for redundancy and to protect the test from impact of mutations. Now, what is a mutation? Just kind of a little refresher for everyone. A mutation can be a substitution wherein one or more nucleic acids are interchanged, switched. It can be an insertion where one or more nucleic acids are inserted into the viral genome. It can be a deletion where they're taken away. It can be a recombination where pieces will switch. Sections of the viral genome are exchanged. And the mutation rate is a term we use to characterize the speed in which the virus mutates. This is a picture of a mutation wheel that we use to convert common mutations in the nucleic acid form into associated protein changes. Now, why is that important? Just for your general understanding of how the mutants are identified. If you're doing NGS in any kind of manner, you're going to need to be able to switch between the nucleic acid and the amino acid. So in this case, L452R would recognize the genetic change here in the center that causes leucine to become arginine. The N501Y, which is a common mutation, flips asparagine to tyrosine, and so forth. So this is a way that you can look at the names that the CDC and the WHO give to the mutations. They're often based on one or more of these nomenclature standards. So this slide describes the four most common methods used to identify SARS-CoV-2 in clinical specimens. We have RT-PCR, which is reverse transcriptase PCR. It is a thermal cycling process and can be manual or automated, and typically it is in a real-time fluorescently tagged type of method. The TMA, transcription-mediated amplification, is isothermal and also automated, and takes advantage of fluorescence in the form of RLUs, or relative light units. The RT lamp is loop-mediated amplification for RNA viruses. It is isothermal. It can be manual or semi-automated. And then the CRISPR, or CRISPR-Cas as some people call it, the clustered regularly interspersed short palindromic repeats, is manual or semi-automated, also isothermal. Why do we use the clinical diagnostic testing, and how has that expanded during the pandemic? We all have recently taken our clinical and diagnostic testing and confined it to symptomatic testing, but we're now also involved in surveillance testing for health-related events, checking for viral shedding, looking for shedding when people go from the hospital to long-term care, and in some cases, non-healthcare related surveillance for schools, businesses, teams, and pre-travel. So it's no longer, our world is no longer respiratory symptoms defined for SARS-CoV-2. We're also involved in contact testing. So when the Department of Health will have exposures that they are known exposures, we can participate in contact testing and following up, and the public health laboratories as well as us will be involved in epidemiological testing to do sort of blind surveillance on the population. That is mostly a public health surveillance issue, but sometimes clinical laboratories do get involved in research as well. Now, in our testing menus, we need to understand that we need to continue to be vigilant for strain variation that could impact our test performance. So we know that there's many thousands of SARS-CoV-2 variants, and the subtypes are categorized for us into larger groups, sometimes called lineages, clades, variant groupings. And the two main ways to do that are in silico testing, where the laboratory or the assay designer will monitor known mutations and check it against the primer probe regions of their testing and make sure there's no problem with the binding in the primers or probes. And then we have in vitro testing, where actual clinical samples with known strain variants are tested, or unknown strain variants are tested to identify if those are meeting the criteria for a variant for SARS-CoV-2. Now, there's a lot of surveillance going on in the United States and around the globe for the viral mutations. In the United States, about 6 to 7% of the virus isolates are being sequenced. The virus mutates at a rate of about 2 mutations per month. They are mostly single nucleotide polymorphisms. And the CDC puts out a bar graph that shows the recent variants and the changes in them. And so you can see here the B.1.1.7 that gained in popularity. And these are available usually on the state and federal public health websites. This is on the right is a map of the vaccination rate. So right now, we're fighting the vaccination rate, hoping it will become higher so these variants don't get out of control. The only way to stop the variants is to get people vaccinated, but there's still a relatively low vaccination rate in certain states and certain geographical pockets, and that is why we have to continue to be vigilant for the viral mutations. CDC classifies the variants and the mutations into 3 major categories. We've talked about all the mutations, thousands of them that have no known consequence, and we won't belabor that any further. But there is a variant of interest, a variant concern, and a variant of high consequence. All three of the variant categories show reduced effectiveness to the monoclonal antibody combinations for therapy and the convalescent and post-vaccination sera. The variant of concern is more severe or more of a worry than the variant of interest. It also has increased transmission, increased severity, as does the variant of high consequence, and a demonstrated failure of diagnostics. So here we come into the range where we are being impacted in the clinical laboratory. And then a variant of high concern, there's really none of these yet, but it would be called if there was reduced effectiveness to any of the therapeutics and medications that are there, or a miss with the vaccine. So these are examples of the SARS-CoV-2 nomenclature and the associated protein changes or amino acid changes. And then up top, you'll see the abbreviations for variants of concern or variants of interest, the VOI. So the alpha variants, the B.1.1.7, originated in the UK. And these are the amino acid changes that you would see in that particular variant. The beta variant is the B.1.351, the South African, similar and other changes there. These are describing some of the common amino acids and how they're characterized, but it's the whole sequence that will actually tell you specifically and confirm that you have these variants in your population. Then we have the gamma variant, the delta variant, the epsilon, zeta, eta, iota, and kappa. So many of these are, you'll see some of them are listed more than twice. The one that's originated in Brazil has the P1 and the P2, and they are named differently because of their genetic dissimilarity. So here you have the WHO nomenclature, which is the Greek. You have the next strain nomenclature, which is the amino acids, and you have the Pango lineage, which is commonly used to identify those strains. So this will give you a little bit of a help when you're trying to sort out which variants they're talking about. It's a little complicated because there's more than one name for each of these. Here are these are the variants of concern, and these are the variants of interest, just listed for you so that you know what they are. The P1, the B.1, 427 and 429, the Delta variant, which we hear about in the news from India, the Beta, and the Alpha. And only the Alpha, the B.1.1.7, originating in the UK, has a gene dropout that actually will cause a failure of some of the targets, but it doesn't cause failure in all of the targets. So it's not still considered an assay that has to be discarded. It can still be used because there's 3 targets in some of the assays that use this particular target. Here's the 3 main nomenclatures that are used when you're sequencing viruses and they're naming viruses. The GISAID is the Global Initiative on Sharing All Influenza Data, and it has been adapted for COVID, SARS-CoV-2. It designates 8 global clades. The next strain has 11 major clades, and that is more for real-time tracking. And then the pangolin, phylogenetic assessment of named global outbreak lineages, this is the one that you'll see that we've been talking about, the B.1.1.7. and others. So those are the major lineage definers, and they're all different genomic databases, and they all have different nomenclature namings. Public health institutes can also institute their own nomenclature changes to track specific variances. And in this country, in the United States, the Center for Disease Control has something called a microbe trace, where they can map person-to-person contacts and pathogen genetic distance and person-to-person exposures. You'll see maps like this on the news, or if you search the WHO site or the Next Strain site, and it will give you the proportion of different variants that are present in different countries across the globe, and it will give you the phylogenetic tree, which tells you the relatedness of the viruses. The shorter the arms, the more related the viruses are. The longer the arms, the more unrelated they are. And this is a CDC website that they will let you sign up for when they have new information. You can sign up to this and get emails about the epidemiology of SARS-CoV-2. So what is the diagnostic and bioindustry surveillance approach? Well, actually, many companies and many laboratories do both in silico and in vitro. And the FDA also monitors the genomic changes in silico, and this is their website for evaluating the viral mutations and the impact on COVID tests. So I would check in with this often, because if there are other variants that impact your test, they will be posted here. Bioindustry generally will at first do an in silico assessment. And this is on the table on the right, just shows a variety of different manufacturers and what their choice of genes are. You can see some have 3, some have 1, and most have 2. So this is where the industry really helps the clinical laboratories actively monitor the genetic reports, and they will announce any information that impacts their assay. The laboratory surveillance approach is in silico if you've designed your own primers, and in vitro for clinical samples that have unusual characteristics. And people are asking, well, how do we pay for this? Why do we need to do this? Do we need to do it at all? So this is one algorithm that we've defined that was designed to try to enrich for the likelihood of us finding variants in our population. The concepts to consider when you're trying to do next generation sequencing or send to a reference laboratory for NGS or use a multiplex for strain identification is the cost, the expected turnaround time, the needs of your local epidemiology, which populations to test, and whether or not to use the NGS or the RT-PCR multiplex panels for variant detection. So in our hospital, we look at any kind of potential outbreaks that might be occurring among people who are fully vaccinated. We look at people in the ICU who are fully vaccinated. Luckily, we haven't had any outbreaks in a fully vaccinated population yet. But we also monitor for unusual test results. And then we form an interdisciplinary review group with laboratory, infectious disease, and infection prevention to determine whether this needs to be investigated. And then the laboratory will decide the best approach, whether to investigate the sample on another platform, to perform the multiplex target assays, or the full NGS. Now this is a pie chart of what we saw this last April of 2021, when we did just a simple 20 samples screened by NGS, and we know that in April the year past, we were 100% wild type, which is up here with the Pango A and B. And now this April, we've gone from 20%, we've gone down to 20% wild type and 80% variants of some kind. Two are not variants of interest or concern, but we were able to identify a proportion, 40% of the B.1.1.7, 20% of the B1526, and 6% of the B1427. So we were able to confirm for our epidemiologists that we have these variants of concern in our population, and we will likely just perform these surveillance from time to time to be monitoring for the Delta variant and such. And if we have any kind of unusual events, we will also do that. There's no diagnostic reimbursement yet, so the testing is really for outbreak investigation only. You can screen for these variants with the multiplex RT-PCR with or without mass spectrometry, the RT-PCR multiplex direct from sample, and NGS. When you're doing considerations for banking or testing your samples, you may want to consider RNA preservatives, using dedicated pipettes and supplies, having an RNase free zone, and extraction and then freezing, perhaps with a protectant. And just a tip there: if you're freezing a high density isolate, you can fill the dead space in the top of the tube with liquid and probably still have enough to keep a cycle threshold that's reasonable. Depends on your CT value, but that dead space can cause water hydrolysis over time, so just be careful of that. Now, the diagnostic methods for the variant testing are to suspect there's a variant, confirm, and perform active surveillance. These are the commercially available multiplex RT-PCR methods: the cobas SARS-CoV-2 variant set, the Simplexa SARS-CoV-2 Variant Direct, the Biorad CoV-2 variant assay, the Allplex SARS-CoV-2 variant, which is CE-IVD, the BioSearch value panels, and the Agena mass array SARS-CoV-2 variant panel, which is all the others are RUO. And finally, the two platforms for next-gen sequencing, which is used for research or epidemiology, is the Thermo Fisher Ion Torrent or the Illumina COVID-Seq. There are other methods. These are the two most common. And they each have research panels that allow you to identify the full genome sequence of interest for the SARS-CoV-2 virus. So in conclusion, we talked about the common molecular targets used in testing patient samples, the major EUA methods, the potential impact of the variants on our EUA test methods, and different methods for variant detection. I thank you very much for your attention, and have a great day.
Thank you, Dr. Wolk, for that informative presentation. I'd also like to thank LabRoots and our sponsor, DiaSorin, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time to answer today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast will be viewed on demand. LabRoots will alert you via email when it's available for replay. We can encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.